Yeah, good morning, students. In the last class of protein and amino acid chemistry, we have already discussed about the chemistry, like classification of proteins, how the proteins have been classified, and also we have different types of classifications of proteins. How the proteins are classified, maybe mainly four types of classifications are there based on their structure and composition. They have been classified into what are the three classes: the simple lipids, conjugated lipids, and derived lipids. And from nutritional point of view, proteins are of two types, that is complete proteins and incomplete proteins. You know that albumin and casein are first class proteins. They have high biological value and they are complete proteins. And based on the shape, proteins are of globular proteins and fibrous proteins. And these are, and based on the function, also proteins are categorized into various types like hormones, catalytic function, transport proteins, etc. Then coming to the physical properties of the protein. So properties of the protein means the biological activity of a protein. It depends upon the number of and the sequence of the amino acids, and the three-dimensional structure of a protein and the function of a protein. It depends upon its physical and chemical properties of a protein. Now here physical properties are the we know that color, most of the proteins are colorless and tasteless, and they are crystalline in nature. And the proteins, shape of the proteins are, they may be either oval shaped globular proteins or fibrous proteins. And depending upon the number of the amino acids present in the protein, the molecular weight depends. Usually the molecular weight ranges from minimum 40 to 40,000, and here for 4 lakhs 40,000, and the number of amino acids minimum vary from 40 to 40,000. Example is insulin, its molecular weight is 5,700, and albumin molecular weight is 69,000. Now here the solubility, coming to the solubility of the protein, and proteins generally they don't form the true solutions because the molecular size of the proteins ranges from in between 2 nanometers to 200 nanometers. If the solute size is less than 2 nanometers, they can form true solutions. If the solute size is more than 200 nanometers, they can form visible precipitate. But proteins in the solutions, their ranges from in between 2 to 200 nanometers. So instead of forming the true solutions, they can form the colloidal solutions. And we know colloids are of two types, emulsioids and suspensoids. So suspensoids means their stability depends on the charge on the surface of the protein, and emulsioids means the stability of these proteins depends on both the degree of hydration, that is shell of hydration, and the electric charge. And even the other physical properties, isoelectric pH, and we know that the proteins are charged molecules. They may in physiological pH, that is in biological fluids at 7.4, proteins carries either positive charge or negative charge. So because of that charge, as they are the charged molecules, which causes the unequal distribution of the ions across the cell membrane, that is known as donor membrane equilibrium. Okay, I repeat here, at physiological pH, proteins are charged molecules. Okay, they may carry either positive or negative charge. So because of that charge, there is unequal distribution of the ions across the cell membrane. So that explains the donor membrane equilibrium. Okay. Now here each protein has their own isoelectric pH. So that is Pi. So isoelectric pH in turn depends upon the number of ionizable groups which are present on the proteins. Based on the so isoelectric pH of a protein that depends upon the ionizable groups which are present on the surface of the protein. Okay, say at their isoelectric pH, we know that proteins are exist as a dipolar ion, that is Victor ion. So there is equal number of positive and negative. So there is no net charge at isoelectric pH, and the proteins below their isoelectric pH, that means under acidic medium, proteins carries positive charge. They exist as cation. And proteins above their isoelectric pH, that means under alkaline conditions, proteins carries negative charge. Now here, and we know that the isoelectric pH that depends upon the number of ionizable groups at the isoelectric pH, proteins are least soluble and gives maximum precipitation. Example of isoelectric pH is albumin, that is 4.7. Isoelectric pH of the casein is milk protein, casein is 4.6. Now the other physical properties, osmotic pressure, we know that osmotic pressure is the pressure which is exerted by the proteins. So usually proteins are colloids, they are heavier than the water. So what happens, proteins, they may displace the water molecules. So because of that, what will happen, albumin, most important plasma protein is albumin. Albumin displaces the water in the blood, so that creates a relative water deficit. Okay, because of that, which help in the exchange of the fluids in between extracellular and intracellular fluids. As the proteins exerts osmotic pressure, colloidal osmotic pressure, the property helps in exchange of the, means maintaining the water balance, electrolyte balance, or exchange of the fluids between intra and extracellular fluids. Then coming to the next one, as the another properties of the proteins are precipitation reactions of proteins. So precipitation reactions means we know that proteins are charged molecules and with a definite size and shape. Okay, proteins are large molecules with a definite size, shape, and they are charged molecules. The stability of a protein in the solution, that depends upon two factors. So that is charge which is present on the surface, and the next one is the degree of hydration. Okay, see here, the proteins in solution, the stability of a protein in a solution depends upon the two factors, that is charge present on the surface, and the next one is the shell of hydration or the degree of hydration. So what is the degree of hydration or shell of hydration means? So the polar groups of the proteins, so which attract the water molecule. So protein has been surrounded by a certain amount of the water, which is known as shell of hydration. Okay, shell of hydration means here polar groups of the proteins, they attract the water molecule, and the protein has been surrounded by some amount of the water in the solutions. So that is known as degree of hydration or shell of hydration. So that means what you have to understand, in protein solutions, protein-protein interactions are less and protein-water interactions are more. Why protein-protein interactions are less in protein solution? Because as the protein has been surrounded by shell of hydration. So that prevents the formation of precipitation. And we know that proteins and in the biological fluids are the charged molecules, carries either positive charge or negative charge. Now, what is precipitation? So precipitation means any factor or any agent which either neutralize the charge on the protein or decrease the availability of the water. That means decrease the shell of hydration, that would result in the formation of precipitation. Okay, generally in proteins in solutions are they means they proteins solutions are charged molecules, and the next one is they have been surrounded by water. But any factor that either decrease the availability of water to the protein, that means decrease the shell of hydration, or the factor which neutralizes the charge on the surface, that would result in the formation of precipitation. Suppose if the factor neutralizes the charge on the surface, what will happen? Bringing it to their isoelectric pH, means making it the neutral. So at neutral pH, proteins are least soluble, gives the precipitation. Or if the factor which decrease the shell of hydration, so decrease the availability of the water, what will happen? Protein water interactions are going to decrease and protein interactions are going to increase. So because of the aggregation or the aggregation of the protein interactions, aggregation of the protein occurs, that would result in the formation of a precipitation. Okay, not only that, even the precipitating agent have the property of bringing them to isoelectric pH, or it may causes it may causes other process, denaturation. Denaturation means disorganization of the native protein. So protein loses its biological activity. Now precipitation means there are the few precipitating agents are there which causes the precipitation of a protein. What of the properties of a precipitating agent? As either four properties it should have. Either it can decrease the shell of hydration, or it can neutralize the charge on the surface of the protein, or bringing it to isoelectric pH, or which causes denaturation. Because of any one of the property, protein interactions are going to increase, so that causes the aggregation of the proteins that would result in the formation of precipitation. Now here, what is the significance of the precipitation reactions? Means generally proteins, from the mixture of the proteins, we do separate the protein by this precipitation methods. Okay, by this precipitation, generally which help in the separation of the protein from the mixture of the proteins, or generally see here in the estimate to get the protein free filtrate in the biological fluids. So in the blood, plasma, cerebrospinal fluid, that is CSF. So in order to prepare the protein free filtrate, so the precipitating agents have been used to remove the proteins. Now protein free filtrate means the filtrate should not contain protein. That means the proteins which are present in these biological fluids, that is blood, plasma, or CSF, cerebrospinal fluids, their protein content has been removed by this precipitating agents in order to get the protein free filtrate. Now here, in the biological estimation of the few parameters, urea estimation, sugar, creatinine, etc. For the diagnostic purpose, we do estimate these parameters, urea, sugar, creatinine in the blood. But before estimating these parameters in the blood, proteins which are present in the blood has to be removed. Why proteins has to be removed before estimating these parameters, urea, sugar, etc. means as the proteins interfere the estimation of these parameters. So that's what when we estimate the sugar content, or the urea amount, or creatinine amount in the blood sample, the first step to be done is to remove the proteins from the fluid blood samples. So how to remove the proteins means by using this precipitating agents. So these are the significance of precipitation. So significance of precipitation is to prepare the protein free filtrate in biological fluids, and estimation of the urea, sugar, creatinine involves the removal of the proteins by using these precipitating agents, and it also precipitation process help in the separation of the protein from the mixture of proteins. Now here, what are the precipitating agents which causes protein precipitation? So the precipitating agents are salts, precipitation by salts and bringing them to their isoelectric pH, okay, and precipitation by heavy metals, alkaloidal reagents, organic solvents, and exposing to heat. All these are the various precipitating agents which causes the precipitation, which would result in the formation of precipitation. Okay, so that means the what might be the action of these precipitating agents? Either they should have one of the four properties: either decrease the shell of hydration, or it can neutralize the charge on the surface, or it can causes the denaturation. Okay, and these are the denaturation. These are the main these are the precipitating agents, so which would result in the formation of the precipitation. Now let us discuss about each one in detail. Precipitation by the salts, how the salts acts as a precipitating agents. So generally the salt like a neutral salt like ammonium sulfate or sodium sulfate, when we add to the protein solution, they decrease the shell of hydration and protein is precipitated. So this is known as salting out. Now what we are discussing, how the salt like ammonium sulfate, neutral salts like ammonium sulfate and sodium sulfate, how they act as the precipitating agent. So when we add these salt to the protein solution, what will happen? The water molecules which has been surrounded by the protein has been attracted by this salt. So that decreases the shell of hydration. So remove the water molecules from the protein. So that increases the protein-protein interactions would result in the formation of precipitation. So, removal of the proteins, okay, precipitation of the proteins by using the salt like ammonium sulfate is known as salting out. Now here the solubility of a protein, it depends upon the ionic concentration of the medium. So the presence of very small quantities of the salt that will increase the solubility of the protein by decreasing the protein-protein interaction. This one is called salting in. Okay, try to differentiate salting out and salting in. Salting in is here the ammonium sulfate. Here it acts as a precipitating agent. Now, how it acts as a precipitating agent? It decreases the addition of the salt, ammonium sulfate, to the protein solution, decrease the shell of hydration. So that increase the protein-protein interaction leads to the formation of precipitation. This process is known as salting out. Then what is salting in? When you add little amount of the, small amount of the salt to the protein, that increases the solubility of the protein. So that decreases the protein-protein interaction. This process is known as salting in. Now here this salting out process is used for the separation of the specific proteins from the solutions. And here the amount of the salt required to precipitate the protein, that depends upon molecular weight of a protein. Right here you know that ammonium sulfate, it acts as a precipitating agent. How? It decreases or it removes the shell of hydration. But how much amount of the salt needed, required to precipitate the protein, that in turn depends upon the molecular size of a protein. Low molecular weight proteins, because of the large surface area, they require more amount of the salt for precipitation. Okay, and high molecular weight proteins, because of the less surface area, they require less amount of the salt for precipitation. So, high molecular weight proteins has been precipitated by half saturation with the ammonium sulfate. And low molecular weight proteins like albumin requires more salt for the precipitation. So, they have been separated from the solution by full saturation with the ammonium sulfate. Then what is this half saturation and full saturation? Means here there are two tests are there. Half saturation test with the ammonium sulfate precipitation. Okay, by salting out, there are two tests are there. Half saturation test with the ammonium sulfate, and the second one is full saturation test with the ammonium sulfate. Right here. And what is the difference between the half saturation and full saturation? Means here in half saturation, we are taking the equal amount of the saturated ammonium sulfate with the protein solution. That means you are taking 3 ml of the saturated ammonium sulfate, means add equal amount, means 3 ml of the protein solution, make the system half diluted. Right here, concentrated saturated ammonium sulfate you are taking 5 ml. Same 5 ml you are adding the protein solution to make the system half saturated. Okay, then filter the solution, and then in the filtrate, okay, filter the solution. In the filtrate, if the proteins are there, okay, in the filtrate, if the proteins are there, means protein is not removed, not precipitated by half saturation. That means the amount of the salt you have added is not sufficient to remove the protein. Okay, and if the in half saturation only, if the filtrate does not contain protein, that means the protein has been already precipitated. That indicates the amount of the salt you have added is sufficient to remove the protein. Right here in the full saturation test, what you do, take the 5 ml of the protein solution, you are only saturating that 5 ml of that one with the adding sufficient amount of the ammonium sulfate. Okay, then filter the solution. If the filtrate has protein, okay, if the filtrate has protein means even that protein is not precipitated by full saturation. Even if that filtrate has, okay, if there is no protein in that filtrate, so that means the amount of the salt you have added in the full saturation is enough to remove the protein. So again I repeat, how the amount of the salt needed to remove the protein means depends upon its molecular weight. Low molecular weight proteins need more amount of the salt for precipitation. High molecular weight proteins requires less salt for the precipitation. Now I have a question for you. How low molecular weight proteins are precipitated? By half saturation or full saturation? Low molecular weight proteins like albumin is precipitated by full saturation with the ammonium sulfate. And high molecular weight proteins like globulins, gelatin, peptones, they have been separated by half saturation with the ammonium sulfate. So, salting out process is used to separate the proteins from their solutions. Okay, albumin is being a low molecular weight protein, separated from the solution by full saturation with the ammonium sulfate. High molecular weight proteins like gelatin, peptone, they have been separated from the solutions by half saturation with the ammonium sulfate. You just remember, less salt is there in the half saturation. Less salt is required for high molecular weight precipitation, protein precipitation, and more salt is there in full saturation test. So more salt is required for the precipitation of low molecular weight proteins. Okay, so this property, salting out property, is used in the separation of albumins from the globulins. Both albumin and globulins are globular proteins, important plasma proteins. So albumin is precipitated by full saturation with ammonium sulfate, and globulin is precipitated by half saturation with the ammonium sulfate. Okay, then here the amount of the salt required in turn depends upon the molecular weight of the protein. There is an inverse relationship. Inverse relationship means low molecular weight proteins need more salt for the precipitation. High molecular weight proteins requires low salt for the precipitation. And how ammonium sulfate, it acts as a precipitating agent by decreasing the shell of hydration and increase the protein-protein interaction that would result in the formation of precipitation. Okay, then coming to the next one, precipitation by organic solvent. Organic solvent like alcohol, even alcohol, it acts as a precipitating agent. How alcohol acts as a precipitating agent means alcohols decreases the shell of hydration. Okay, because alcohols are dehydrating agent. So when organic solvent like alcohol is added to the protein solution, being alcohols are dehydrating agent, they decrease the availability of the water to the protein and increase the protein-protein interactions that would result in the formation of precipitation. Okay, so here how the organic solvents means, when the organic solvent is added to the protein solution, water molecules which are available for the proteins are reduced, since organic solvents are dehydrating agents, and that causes precipitation. Examples are ether and alcohols are ethyl alcohol, methyl alcohol are the powerful precipitating agent. And what is the significance of this precipitation by organic solvents means, see here, surgical spirit we do use after phlebotomy, means after veiny puncture, after the blood sample collection, we do rub the site of the puncture, vein puncture by applying the surgical spirit. Why means here the surgical spirit which contains alcohol, it acts as a disinfectant. How it acts as a disinfectant means because of the property of precipitating proteins. Okay, like that here this is the organic solvent. It acts as a precipitating agent. Its significance is surgical spirit is used during surgery or after the blood sample collection. It acts as the disinfectant because of its property of precipitating the proteins. Then coming to the next one, precipitation by heavy metal ions. Okay, heavy metal ions means alkali reagents. They act as precipitating agents. Example, salts of the iron, copper, zinc, lead, cadmium. All these salts, when you add into the protein solution, that causes precipitation. How in the presence of these alkaloidal reagents, heavy metals, how these proteins are precipitated means, here proteins are negatively charged in alkaline medium. Okay, presence of these heavy metal ions which makes the medium alkaline. Under alkaline medium we know that proteins are negatively charged, so the positively charged metal ions, they complex with the they complex with the positively charged metal ions. They complex with the negatively charged these proteins, and that would result in the formation of precipitation. Okay, see here how the heavy metal ions acts as the precipitating agents. Here means when you add heavy metals into the protein solution, the proteins are negatively charged in alkaline medium. Okay, and the positively charged these metal ions, they neutralize the negatively charged proteins and causes the precipitation. And mercury is toxic to the toxic. So the toxicity of the mercury is when you ingest, they causes the precipitation of the GIT proteins. Okay, so that's what the mercury is toxic. And the raw egg is used as the antidote in mercury poisoning. So what is the action of the raw egg means? The raw egg contains albumin that may combines with the mercuric salt to form a mercury proteinate, and that prevents the absorption of the mercury. So here based on this property, so mercury is toxic to the body as they precipitate the proteins of the GIT. And here the antidote which will be given for the mercury poisoning is the raw egg, because as the raw egg contains the protein albumin that combines with the mercuric salt and forms the insoluble mercury proteinate, and that in turn decreases the absorption of the mercury, thereby decreases the toxic symptoms associated with the mercury poisoning. Then coming to the next one is here precipitation by the see here it's a precipitation by the heavy metal ions. Then the next one is the precipitation by the alkaloidal reagents. So alkaloidal reagents means acids like picric acid, phosphotungstic acid, sulfosalicylic acid, trichloroacetic acid, that is TCA. All these are the alkaloidal reagents. They act as the precipitating agents. So how they act as the precipitating agents means the presence of these alkaloid reagents makes the medium acidic, and we know that the acidic pH, okay, that means the proteins below their isoelectric pH, they are positively charged. Okay, so pH below their isoelectric pH, that means under acidic medium, proteins carry the positive charge, and the negative charge of these alkaloid reagents neutralize the positive charge on the protein and causing the precipitation. Now here, how the alkali reagents, they act as precipitating agents means only 2 points have to remember. The presence of alkaloidal reagents makes the medium acidic. Under acidic conditions, proteins carries positive charge. So the negative charge of alkali reagents neutralize the positive charge of proteins and bringing and causes the formation of the precipitation. Example here, the TCA, trichloroacetic acid is used, one of the precipitating agent in the preparation of the protein-free filtrate when you estimate the biochemical parameters. I already mentioned in the previous slide, in the estimation of the few biochemical parameters, proteins need to be separated prior to the estimation as the proteins interfere the estimation. So how the proteins has been removed from the biological fluids means by using the alkali reagent, that is TCA. TCA stands for trichloroacetic acid. And the next one is sulfosalicic acid is one of the alkali reagent. It precipitates the protein. It is widely used in the quantitative estimation of the proteins in the urine. Okay, so protein quantitative estimation, the amount of the protein which is there in the urine has been estimated by using this alkaloidal reagent, that is sulfosalicic acid. And the next one is isoelectric precipitation. So that means you know that precipitation is the precipitating agent. It should have one of the property, either bringing into isoelectric pH or neutralize the charge or decrease the availability of the water, or it causes the denaturation. Now coming to the isoelectric precipitation means all bringing it to the few precipitating agents when you add to the protein solution which bring the isoelectric pH of the particular protein. So bringing it to isoelectric pH also causes the formation of precipitation. And we know that all proteins at their isoelectric pH, that is Pi, they are least soluble because at the isoelectric pH, proteins exist as Zwitter ion, dipolar ion, where it has equal number of positive and negative charges. So at the isoelectric pH, proteins are least soluble and gives maximum precipitation. So this property, isoelectric precipitation property, is used for the separation of the proteins from the mixture. Okay, from the mixture of the protein, few proteins has been separated based on their isoelectric pH. For example, you take casein. Casein is the milk protein. Its isoelectric pH is 4.6. Okay, that means it forms a precipitate at 4.6 and redissolves in acidic or alkaline solutions. Okay, the pH of the casein is 4.6 or okay, means at 4.6 casein is precipitated and the precipitate is redissolved. Either you decrease the pH bringing it to acidic pH or else increase the pH or bringing it to alkaline pH redissolves the protein precipitation, that is casein. Now here, this property is used in the curdling of the milk because when milk is curdled, how the milk is curdled means the bacterial fermentation of the curd releases lactic acid. Okay, milk contain protein casein. You know that isoelectric pH of the casein is 4.6. So because when you add curd into the milk, what will happen? The bacterial fermentation of the curd causes the release of lactic acid. Being an acid, this lactic acid decreases the pH of the milk, bringing it to isoelectric pH of the casein 4.6. Okay, when you add acid to the solution, as the acids are hydrogen donors, increase hydrogen ion concentration decreases the pH. So lactic acid, which has been produced by the bacterial fermentation of the curd, decreases the pH of the milk, bringing it to isoelectric pH of the milk protein casein, that is 4.6. So curdling happens. So because at the 4.6 casein is precipitated, so that causes the curdling of the milk. Okay, so here this is the isoelectric precipitation. Now here the next one is precipitation by heat coagulation. Heat coagulation means here even applying heat to the protein is also one of the precipitating agent. Okay, on heating, heat coagulable proteins like albumin globulins, they may undergo denaturation. Okay, especially not all the proteins. Heat coagulable proteins, example, albumin and globulin, when exposed to the heat, they undergo a process known as denaturation. So, what is denaturation? Disorganization of the native protein. That means, so the primary structure means the amino acid sequence is intact, but their changes will be seen in the secondary, tertiary and quaternary structure of a protein. Okay, when heat coagulable protein exposes to the heat, they undergo denaturation. During denaturation, the primary structure is intact. Primary structure means this will not disrupt, change the amino acid sequence, but changes will be observed in the secondary, tertiary and quaternary structure of a protein. Because of that, protein loses there is a changes observed in physical and chemical properties of the protein. Because of that, these proteins when exposed to the heat causes the formation of precipitation. Okay, white PPT is observed. The turbidity, precipitation is nothing but a turbidity, or that is okay. So on heating, some proteins like albumin are denatured. Denatured lose their structure to form a white turbidity or white PPT. When you apply heat to the turbidity, which forms a coagulum. Okay, and now here this heat coagulation, this property, precipitation by heat coagulation property is mainly used in the heat coagulation test. Okay, this is the heat significance of the heat coagulation test is this is the test used to detect proteinuria. Okay, proteinuria means presence of proteins in the urine. Generally, proteins will not excrete in the urine. Presence of the protein in the urine is an abnormal condition. Under pathological conditions only, protein started excreted through urine. So presence of proteins in the urine is known as proteinuria. Okay, so the proteinuria is detected by simple test, that is heat coagulation test. Heat coagulation test based on the principle precipitation by the heat coagulation. I think you are following. Heat coagulation test is one of the basic tests to be performed in the clinical laboratory to detect proteinuria. Proteinuria is nothing but proteins in the urine. Proteinuria is the term denotes proteins in the urine. Under normal healthy conditions, proteins, okay, there is no excretion of the proteins in the urine. Only in the pathological abnormal conditions only proteins are started excreted through urine. So proteinuria can be detected by the test heat coagulation test. So how to do the heat coagulation test means simple test. There you have to take the urine solution in a test tube and boil only the upper part for 2 minutes. Okay, you need to fill 3/4 of the test tube with the given urine sample. Okay, and boil the upper half for 1 minute. So if albumin and globulin, if albumins are there in that sample, when exposed to the heat, they undergo denaturation and result in the formation of white PPT. On boiling for 1 or 2 minutes in the heated portion, white PPT is observed if that urine has protein, that is albumin, because albumin is heat coagulable protein. When exposed to the heat, denatures to form a white PPT. Then add few drops of acetic acid. Why we are adding acetic acid after precipitation means bringing it to isoelectric pH. Isoelectric pH of the albumin is 4.7. When you add when you add acetic acid into that, decreases the pH, bringing, if albumin is there, if it reaches 4.7, you will get more precipitation. So addition of acetic acid provides the isoelectric pH to get the maximum precipitate. And there is one more reason is there to add acetic acid, that is acetic acid also dissolve the turbidity which is formed due to the phosphates in the urine. Okay, now here heat coagulation test is the test done to detect proteins in the urine. How to do the heat coagulation test? Just fill 3/4 of your test tube with the given provided urine solution. Boil the upper half for 1 minute. Then add few drops of 1% acetic acid. If white coagulum is there in the heated portion, that indicates heat coagulation test is positive. Test is positive means that indicates proteinuria. That means proteins are there in that given urine sample. So what is the principle behind this one? If albumin is there, when exposed to the heat, it undergoes denaturation. We know that denaturation is the disorganization of the native protein. Okay, primary structure is intact, but there is a changes observed in the physical and chemical properties. So, protein loses its biological activity, so that would result in the formation of precipitation. Okay, then why you are adding acetic acid to that precipitation means two reasons are there. Because the normal urine contains phosphates. Sometimes phosphates also, when exposed to the heat, precipitates, okay, and form white PPT. We don't know whether the precipitation is because of the phosphates or because of the albumin. So in order to differentiate that, we do add acetic acid, because if you add acetic acid, bringing it to isoelectric pH of the albumin 4.7, the precipitation intensifies. You will get the maximum precipitation. Okay, and another reason to add acetic acid is it dissolves the turbidity if it is formed due to the phosphates. Now, by this, based on this principle, proteinuria can be detected by simple test, heat coagulation test, and another test which will detect the proteins in the urine is sulfosalicylic acid test and Hurler's test. Okay, that's it about the precipitation reaction. Okay, precipitation means here any factor utilize the charge, decrease the shell of hydration, bringing it to isoelectric pH, or exposed to the heat causes the formation of precipitation. Already in the previous slides we have discussed about isoelectric precipitation, how the precipitation by heat coagulation, precipitation by alkali reagents, precipitation by heavy metals, precipitation by using the organic solvents, then precipitation by using the salts like ammonium sulfate. And all these precipitating methods are used for separation of the proteins. Now here coming to the next one is color reactions of the proteins. So color reactions of the proteins means we know that proteins are nothing but polypeptide chains made up of amino acids, and we know that amino acids, the amino acids differ in their structure at their side chains. So because of the presence of the functional groups of the amino acids, okay, or because of the presence of peptide bonds, proteins may react with the various reagents and form color. So that's what these reactions are called, color reactions of the proteins. Till now what we have discussed in the previous slides, those are the precipitating agents, how those addition of those precipitating agents causes the formation of precipitation. Now what we are discussing, color reactions of the proteins. So color reactions means proteins when they react with the different reagents, okay, different chemicals, they will give different colors. Why different colors has been formed when proteins interact with the various chemicals means because of the presence of the functional groups or because of the presence of the peptide bonds, proteins may react with the various chemical agents and form color reactions. So these are the examples of the color reactions. The first one is the bio-red test. Biuret test is the simple basic test to detect the proteins. Okay, so biuret test is the basic test from the unknown solutions. How do you separate the protein solutions? Means by performing the simple test, that is biuret test. Okay, and biuret test detects the presence of peptide bonds. Peptide bonds are present between two amino acids. And the next here the test color of the biuret test is a purple color is observed. You can take some unknown solution, add NaOH, few drops of NaOH and copper sulfate, mix and see the color. If purple color is observed, that indicates the presence of peptide bonds, indicates the presence of protein. Then the next one is ninhydrin test. Here also take the protein solution, add ninhydrin solution and boil for 2 minutes. If purple color is observed, that indicates the presence of alpha amino acid. Now the another color reaction test is the xanthoprotein test. Here also in the xanthoprotein test, there is a orange colored PPT is observed. That indicates the presence of aromatic amino acids. So how to do the xanthoprotein test? Just take the protein solution, add HNO3 nitric acid, boil for 1 minute, then add alkali NaOH. Okay, if orange colored PPT is observed, that indicates the presence of aromatic amino acids like phenylalanine, tyrosine and tryptophan. Next, another test is their color reactions, Milan's test. The Milan's test, light red color PPT is observed. That indicates the presence of the amino acid tyrosine. Then aldehyde test. Here in the aldehyde test, there is a purple ring is observed. Okay, that indicates the presence of indole group containing amino acid tryptophan. Next, another color reaction test, Sakaguchi test. Here in the Sakaguchi test, dark red color, deep red color, cherry red color is observed. That indicates the presence of guanidino group containing amino acid arginine. Okay. The next color reaction test is sulfur test. If black colored PPT is observed, that indicates the presence of sulfur containing amino acid cysteine and cysteine. Anyway, these details, how to do the test, procedures, principles, color reactions, okay, procedure, principles, observation, inference, details will be given in detail in practical lab. Okay, here in the theory class, just I am giving you the list of the color reactions and their significance. Details will be discussed in detail in the practical class. So like that, these are the properties of the protein, like physical properties and the precipitation reactions of the protein and color reactions of the protein. Okay, the next coming class we will discuss about the structural organization of the protein. So how the proteins how they will undergo, so proteins will undergo 4 levels of structural organization okay, that is primary, secondary, tertiary and quaternary, and what are the bonds which will stabilize the protein structure, and how the proteins will undergo 4 levels of organization. That will be discussed in the next class. Thank you.